So in this video presentation, we are going to learn about the formation of goiter. Now goiter formation can take place in either of the condition, in hypothyroidism as well as in hypothyroidism. Whether it is increased amount of T3, T4 is there, or decreased amount of T3 and T4 is there, due to any cause, goiter formation can take place. Goiter is nothing but enlargement of the thyroid gland. So this is the follicular cell by which the T3 and T4 hormone is being produced. Okay, in detailed synthesis and the secretion of the hormones has been dealt in another video. This is a very concise form. I am going to tell you that iodine is being trapped by the sodium iodide sympater. Okay, it has been taken up through the pendant molecules to the colloid. This iodine form has been oxidized. Okay, so there is the organification of the iodine ion is there. It gets attached to thyroglobulin molecules which is being formed in the cells, and when one iodine ion being attached, it is known as MIT. If two is being attached, it is known as DIT. 3 become T3, and when 4 iodine attached become T4. Now it has been endocytosis, and by lysozyme enzyme there is a proteolysis mechanism that take place. As a result of that, T3 and T4 has been released into the circulation, and MIT and DIT, in which the iodine ion has been attached, is again being recycled for the resynthesis of the thyroid hormone. So goiter formation can take place either in hypothyroidism or in hyperthyroidism. When T3, T4 production is there, a condition is known as the myxedema, which happen in the adult. And when there is a decreased amount of T3 and T4 is there in the childhood period, it is known as the cretinism. Now grave disease, it happen in the adult. As a result of that, what is happening? There is a increased amount of T3 and T4 production is there, and it is being grave disease. The name is being given as grave because of the scientist Robert James Graves, who described a case of goiter with exophthalmos in 1835. Now normally what is happening? Thyroid releasing hormone is being secreted from the hypothalamus. It goes to the anterior pituitary in the thyrotroph cells. From the thyrotroph cell, thyroid stimulating hormone has been secreted, and it goes through blood to the thyroid gland. Okay, in the thyroid gland, it stimulates the thyroid gland to synthesize the T3 and T4. Okay, now when amount of T3 and T4 is less, okay, so negatively in normal circumstances is going to feedback to, it will be given to the hypothalamus and the anterior pituitary. This is the negative feed it is going to give, that there is a less amount of T3 and T4 is in the blood, so please try to increase the amount of T3 and T4 in the blood. So by negative feedback, okay, the amount of T3, T4 will be increased. Less amount, it will give a signal to the hypothalamus to increase amount of TRH, and TRH in return is going to stimulate the anterior pituitary to increase the TSH, and again TSH will stimulate the thyroid gland to produce the T3 and T4. Okay. Now what will happen if there is a lack of iodine in the diet, or if you are taking less amount of iodine? Now we know that for the synthesis of T3 and T4, two raw material is required. One is the iodine, and the second one is the thyroglobulin molecules within the follicular cell. So if there is a deficiency of one iodine are there. Okay, so what will happen if iodine is not there? So in the synthesis of the iodine, what is happening? The NIS is taking less amount of iodine, less amount of oxidation will take place, less amount of coupling, and less amount of everything is going to happen within the follicular cell, and as a result of that, less amount of T3 and T4 will be formed. It is going to produce a very little amount because already there is a lack of iodine is there. Okay, So lack of iodine is going to produce, one raw material is missing, so it is going to produce very little amount of T3 and T4. Okay, so now when there is a very little amount of T3 and T4 is being produced, okay, so it is not going to negatively inhibit, negatively feedback will not be given to the hypothalamus or the anterior pituitary. So there is a lack of negative feedback inhibition. Now as a result of that, if negative feedback is not given over here, so, okay, so what will happen? The hypothalamus will produce large amount of thyroid stimulating hormone. Okay, in response to that, anterior pituitary is going to produce large amount of thyroid stimulating hormone. Okay, and this thyroid stimulating hormone is going to stimulate the gland to produce more amount of T3 and T4. Okay, but since the raw material is missing, okay, again gland will not be able to synthesize T3 and T4. But since there is the stimulation of the thyroid gland by the thyroid stimulating hormone, so what is happening within the cell? The NIS, sodium iodide transporter, will be not taking up the iodine ion tissue deficiency. Okay, and secondly, since the virtue of the gland which is being stimulated by TSH, large amount of thyroglobulin is going to be formed within the cells. Now these thyroglobulin molecules will be accumulated in the colloid, okay, and it will not be used. So lot of accumulation of this colloid material will make the gland enlarge, okay, and goiter. Okay. Now what will happen if there is a stimulation of the anterior pituitary by the some another substance other than the thyroid stimulating hormone? Okay. Now we have seen that from the hypothalamus thyroid hormone is being released, okay, normally. Now anterior pituitary will release thyroid stimulating hormone, and this thyroid stimulating hormone is going to stimulate the thyroid gland. Now if instead of thyroid stimulating hormone, some another substance, some other molecule is stimulating the thyroid gland, for example, there are certain immunoglobulins are there, and these, when these immunoglobulins are stimulating the thyroid gland, they become thyroid stimulating immunoglobulins. Okay, so the thyroid stimulating hormone can stimulate the thyroid gland for approximately 1 hour, but these immunoglobulin, these antibodies, thyroid stimulating immunoglobulin, will stimulate this thyroid gland for more than 12 hours. Constantly it is going to stimulate. When this thyroid gland is being constantly stimulated by these antibodies for a very long period of time, what is going to be happen? There is a hyperplasia of the cell is going to happen. Okay, when hyperplasia of the cell, number of cell is going to be increases. Number of cell increases, and by the virtue of the stimulation, okay, what is going to happen? Large amount of T3 and T4 is going to produce. This will negatively feedback the anterior pituitary and the hypothalamus to produce low amount of TRH and TSH. So always in hyperthyroidism you will find low amount of TSH, and in cases of hypothyroidism you are going to find large amount of thyroid stimulating hormone. Okay, so once again I am repeating, in cases of hypothyroidism you are going to find large amount of increased amount TSH and low amount of T3 and T4. In cases of hyperthyroidism you are going to find less amount of TSH and more amount of T3 and T4. Thank you.